Okay, this is a walkthrough of June 2012 and it's the biology B1 and it's higher tier. Okay, so just to remind you again, remember looking at the front of the paper, the main bit we want to focus on is down here, which tells us which question is going to be marked for grammar. So question 3 is going to be answering continuous prose on this paper, and you're going to be marked for your use of good English, organising information clearly and using specialist vocabulary where appropriate, and obviously spelling that right as well. Okay, the rest of it is just the general information using the black or blue ink, and it's out of 60, etc. Okay, remember you can use a calculator for all exams, including biology. Please make sure you take it into the exam with you. Right, so let's start by looking at question 1. So question 1 states that scientists at a drug company developed a new pain-killing drug, drug X. Painkillers do not cure infectious diseases. Why? Well, a painkiller doesn't kill the pathogen, okay? So it, that's not very good English, is it? Doesn't kill the pathogen, okay? You could have said bacteria or virus, etc. But the idea is a painkiller doesn't kill anything, it just helps relieve the symptoms, okay? Right, let's carry on with the question. The scientist compared drug X with 2 other pain-killing drugs, drug A and drug B. In their investigation, the scientist chose 600 volunteers, the volunteers were all in pain, gave 200 of the volunteers a standard dose of drug A, gave 200 of the volunteers a standard dose of drug B, and gave 200 of the volunteers a standard dose of drug X. Over the next 7 hours, the volunteers recorded how much pain they felt. To get valid results, so this V, think variables, to get valid results, the 3 groups of volunteers should be matched for as many factors as possible. Suggest 2 of the factors that should be matched. So what this is saying is, what are your control variables? What things we want to keep the same in all of these people, okay, to make sure it's a fair test. So things that would be good would be things like your age, the gender, so male or female, how much pain they're in, because if someone's in loads of pain, someone's in not a lot of pain, that wouldn't really be very fair. You could have then for the type of illness, their weight or their mass, their body mass, okay? So if we put age, that would be fine as well, and we could have amount of pain they had before trial. Okay, so there's a good answer for that one. Right, on to the graph question then. 1C. The graph shows the results of the investigation. So let's look at this first before we go on to the question. The mean percentage decrease in pain here, so this is how much they're decreasing in pain. The time in hours after taking the drug. So we can see that after taking the drug, after 1 hour, all of them have shown a decrease in pain. Drug X showing the most decrease in pain, A being the lowest and B sitting somewhere in the middle. So let's look at the question it's going to ask us about. So how much pain did the volunteers still feel 4 hours after taking drug A? So let's check this on after 4 hours. So after 4 hours of taking drug A, and we read across there to here, their mean decrease in pain, percentage decrease is 25%, okay? So if they had 100% and they've decreased by 25%, they're still going to feel, okay, 75%. And that caught out an awful lot of students in the year this paper was sat. They all wrote 25 because they read the number straight off the graph and did not read the axes. Give 1 advantage of taking drug A and not taking drug B, okay? So what's 1 advantage of taking drug A and not drug B? So if we compare drug A and drug B, we can see that drug A seemed to act faster, didn't it? Because if you see here, it's hit the pain relief after 1.5 hours at sort of about 65%, 70% pain relief. Whereas drug B took nearly 3 hours to reach its peak. So it was certainly faster. Drug A gave faster pain relief. So I'll put that one in here. Okay, so faster pain relief. Right, give 2 advantages of taking drug B and not drug A. So the alternative way round. So let's have a look. 2 advantages of taking drug B and not drug A. So let's have a look at this one. Well, drug A falls away really quite quickly. So after after sort of 2 hours, you're now getting an increase in pain again because the decrease in pain is dropping away. So they're going to start to feel more pain. So their decrease in pain doesn't last very long, whereas drug B lasts much longer. Okay, so and it also eventually, even though it takes longer, does get to a higher decrease in pain, whereas drug A only gets here. Drug B is reaching a decrease in pain somewhere over there. So B does give them more pain relief, and it lasts much longer. Okay, so 2 advantage is the decrease in pain is higher. Okay, and the other point is the decrease in pain lasts longer. Okay, so that's the 2 advantages there. Again, I've just bullet pointed these. I've got no marks or paragraphs. It's asked me for 2 in bold. I've given 2 clear statements. Okay, drug X is much more expensive than A and B. A pharmacist advised a customer that it would be as good to take drug A and drug B together instead of drug X. Do you agree with their advice? Give your reasons for your answer. Now we need 3 marks here, so we need 3 clear things. Now we could give fors and against, absolutely fine. We're not going to get a mark for yes or no, so you could give pros and cons if you couldn't think of 3 straight away things. So let's have a look at drug X. What are the advantages? Well, the advantages are, again, we get almost 100% pain relief with drug X, so that would probably be a good reason. And again, it's pretty rapid. It follows the same line as drug A and then keeps going. So, if you have a look here, when it's reached its peak, it's still before the peak on B. So it gives more pain relief and it does it much faster. Okay, so that would be quite useful. But we do know that it costs quite a lot more money, so that would be a negative, wouldn't it? We could also think that it's not a great idea because if we take 2 drugs together, could they have a problem? Could they have a side effect? Could this person overdose? Okay, so again, that's probably something we could give an argument against. So the drug X is more expensive than drug A and B. A pharmacist advised a customer that it would be just as good to take drug A and B together instead of drug X. Do you agree with the pharmacist's advice? Okay, so we're going to say we don't agree with A and B. No, because taking 2 drugs together may interact with each other and they may overdose. So that's 2 marks there. The idea of interacting and the idea of an overdose would get a mark. Okay. However, okay, A and B, what we're going to say is positive about those. So A and B cost less than X, and drug B gives pretty long lasting pain relief compared to X. So that is a slight benefit there in B. We can get the rapidness from A and the long lasting bit from B. So it almost covers it for the pharmacist. Okay. So however, A and B cost less than X, and A provides rapid pain relief and B provides long lasting pain relief. Okay, so both points there, so we can ensure we get all 3 marks for that question. Okay, right, let's go on to question 2. Okay, question 2. The diagram shows part of the carbon cycle in a habitat. So here we can see the cycling of carbon around and the arrow showing how carbon is moving. Name the processes shown by arrows X and Y. Okay, so let's have a look at X. X is going from the microorganisms up to the atmosphere. So what are microorganisms doing that's releasing carbon into the atmosphere? Well, they are respiring. So respiration, okay, a process normally ends in T-I-O-N, like respiration, okay? Photosynthesis is an exception to that rule, but as a clue, if asked for a process, it's asking for something ending in T-I-O-N. Right, Y is, if we have a look on here, fossil fuels going into the atmosphere. We know that when we burn fossil fuels, it releases carbon dioxide into the atmosphere. Now, you would get away with burning, but the better word would be combustion. Again, ending in T-I-O-N. Right, describe the part played by algae in this carbon cycle. So algae are giving carbon dioxide to the atmosphere. Well, how are they doing that? How are they producing carbon dioxide? Well, this bit would be respiration. So I'm going to just label the arrows so I know my answer. They're taking in carbon dioxide from the atmosphere, so doing that in photosynthesis. Okay, they're a green plant. And they're also releasing something over here into the microorganisms. So what's happening there? Well, they're being fed on, or they're decayed by the microorganisms. That's decay going around there. So 3 things, and it's worth 3 marks. So we're just going to write a little bit about each of these processes for this section here. So describe the part played by algae in the carbon cycle. It takes in carbon dioxide in photosynthesis. It releases carbon dioxide in respiration, and when carbon is passed on to animals. So here it's being eaten. And passed on to microorganisms when they decay it. Okay, so we've got lots of good facts there worth the 3 marks. We've talked about photosynthesis, we've talked about respiration, we've talked about it being eaten, we've talked about it decaying. Okay, I think we've covered our backs there in terms of marks. Right, in tropical rainforest, process X is much faster than in most other habitats. Why? So what do microorganisms need? We've said that X is, here we've got respiration. Okay, so in tropical rainforest, process X is much faster than the habitats. So why are microorganisms respiring more in a tropical rainforest? Well, they're obviously doing more decay. And why are they doing more decay? What conditions do we need for decay? Well, it's warmer. Okay, so rainforest is much warmer, it's hot, and that's what microorganisms like for decay. It's also moist or damp, and again, that's a condition needed for decay. Okay, we've also probably got a lot of microorganisms in a rainforest. That might be a little bit of a guess, but we've also got a lot of material. Okay, so a lot of things to decay down in a rainforest. So a rainforest okay, so why is it much faster? It's because it's warmer, it's got moisture or damp, which both the conditions are needed for decay. So if I put that on there, conditions needed for decay. It's also probably got a lot more material to break down, but that would be an extra mark. Okay, it would be these 2 here for the 2 marks for the question. Right, on to the next bit. So this is the bit where we were told at the very start, this is the one we were going to mark for good English, organising information clearly and using specialist terms where appropriate. So this is where we need to think, have a little pause, plan it out, and then start your question. Plants and animals have become adapted in many different ways to reduce the risk of being eaten by predators. So we are describing the adaptations that reduce the risk of being eaten by predators. Okay, very important. We're not asking how they can survive in dry conditions. It's specifically about how they can not be eaten by predators, and we need to stay with that theme, otherwise we're not going to gain the marks. It also asks us to give examples of animals and plants. So if we only do one, again, we're not going to get the marks here. So let's have a little think about how animals and plants can be adapted to reduce the risk of being eaten by predators. So you might think that some plants have spikes or thorns, and that will prevent predators from eating them as they are sharp. So we want a full sentence, okay? So some plants have spikes that will prevent predators from eating them as they're sharp. So what else can things have? Well, some animals might have camouflage so they can hide, so they aren't eaten by their predators. So some animals may be camouflaged so predators cannot see them to hunt them. Okay, so there's a good couple of points there, but we are going for 6. I think we probably need at least 2 more to get those extra marks. Let's think, what else could animals or plants have in order to prevent predators? Well, if you think about herbivores that are often hunted by carnivores, like cows and sheep, things that we see every day, their eyes are on the side of their head. They have really good field of vision, whereas most carnivores, their eyes are at the front of their heads. They can get good depth perception for hunting. So we could talk about the position of the eyes and also large ears. They can hear their predators coming. So some animals may have eyes at the side of their head to detect or increase their field of view so they can see a predator more easily. Okay, we could also talk about some animals that have a real nasty taste to them, or poison. So some animals really don't rate taste very nice. So the animal eating it might actually spit it out before they kill it, or they're less likely to go and hunt anybody else. Okay, so we can say that again, some animals or plants may have a bad taste or poison which makes the predator spit them out or stop eating them. Okay, so I think we've got enough points there in terms of covering what we've done, but we have mentioned something they've got and why it stops the predators from getting them. So we've explained every single point. We haven't just described the feature, we've explained how it reduces the chance of the predators getting them. Okay, right, on to question 4. Okay, question 4. Charles Darwin investigated tropisms in plants. Some students did an investigation similar to Darwin's investigation. The students grew seeds until short shoots had grown, used black plastic to cover parts of some shoots, Put the shoots in light coming from one direction. Put boxes over the shoots to keep other light out. The diagrams show how it's set up. So A had light coming from one side. B had black plastic round the bottom part with light coming from this side still. And C had black plastic on the top with light coming from one side. So 2 days later, the students took off the black plastic covers and looked at the shoots, and the diagrams show the results. Give 2 variables that the students should control in this investigation. So again, back to sort of similar to question 1, thinking about the control variables to make sure it's a fair test. So it's plants, so think about what sort of things are going to affect plants. So we need to make sure we keep the amount of light they get the same. So light intensity needs to stay the same. Amount of water will affect it as well. But you could also think about, well, it's only worth 2 marks, that would have got 2 marks. But for your benefit, you might also want to think about type of plant or species of plant or seed. And also maybe the age of the plant, okay, or size. So age or size of the shoot. Okay, all good things to consider when doing this sort of practical. Right, shoot A bent towards the light as it grew. Explain how. So let's have a little look at shoot A. So we can see here it's bent toward the direction of the light. And we need to have a think about what's caused that. Now, that's caused by auxin. Okay, and auxin goes away from the light. Okay, so auxin is a hormone, plant hormone, and what it would do is it would collect on the far side of the plant, so over here, and we get an unequal distribution. And that makes this side grow much faster, so it causes it to bend towards the light. Okay, so let's write that down in some words to get those marks. So here we've got, we need to say the hormone auxin controls growth. It collects on the side furthest away from the direction of light. Okay. It causes the side in the dark to grow faster causing the shoot to bend towards the light. Okay, so we've got a good answer there for 4 marks. We've talked about the hormone auxin. We've talked about the fact that it's collecting on one side. A better way for describing it collecting on the side first way from the direction of light or on one side, you could use the term unequal distribution, which I think is quite an important term to use when talking about auxin. In fact, it's unequal. So it collects on the side first way from the light, okay? And so we get more on the dark side. It causes the side in the dark to grow faster, causing the shoot to bend towards the light. Okay, right, on to the step 4C then. What conclusions can be drawn from the results about the detection of the light stimulus? So let's have a look. So this one bent towards the light, this one bent towards the light, and this one grew straight up. So if we look back here at what they did in the experiment, well, this one had the top of it covered. So what it would suggest is that the part of the plant which detects the direction of light is actually in the top of the plant and not in the bottom of the plant. Okay, so what conclusions can be drawn about the results from the detection of the light stimulus? The plant detects the light using the tip or top end of the plant. Where in the shoot the response to the light takes place, So where does it take place in terms of where we're getting the response to light? Well, that's just behind the tip where we're getting the response. Okay, so we're going to say behind the tip of the shoot coming out. All right, good. Let's go on to question 5. Okay, this is question 5. People may be immunised against diseases using vaccines. Which part of the vaccine stimulates the body's defence system? Well, a vaccine is made up of dead or inactive pathogens or bacteria or viruses, whatever it might be. So we're going to put in here dead or inactive pathogens. And that will get us the 2 marks. 1 mark for pathogen, 1 mark for dead or inactive. Okay, a person has been vaccinated against measles. The person comes into contact with the measles pathogen, but the person does not catch measles. Explain why. So if they've already been vaccinated, their body would already have seen the pathogen, okay? So they would already have had the primary response, and now it can just reinitiate that response again. So what's going to happen is the person's white blood cells will produce antibodies. This is the first thing, and it's really important that you remember this term, okay? So the first thing they do is produce antibodies, and they produce them really quickly. That's why you vaccinate, so someone can respond very rapidly. So person's white blood cells produce antibodies rapidly or quickly, okay? And the antibodies destroy the measles pathogen. Okay, so key things, talking about antibodies, talking about it happening rapidly. That's why if we would respond anyway most of the time, but it would be so slow we'd get really quite poorly. So we vaccinate, so we have a rapid response, so you don't even notice you're ill, and the antibodies then destroy the measles pathogen. Okay, on to 5B. A man catches a disease. The man has not been immunised against this disease. A doctor gives the man a course of antibiotics. The graph shows how the number of live disease bacteria in the body change when the man is taking the antibiotics. So here we've got the number of live disease bacteria in the body in billions. So this goes up here. And the time in days from when he started his course of antibiotics. So we can see that from when he took it, they still increased, and then they rapidly decreased over those days. But interestingly, it's not gone to 0 here. Okay, let's look at the questions. So 4 days after starting the course of antibiotics, the man feels well again. It's important that the man does not stop taking antibiotics. Explain why. So let's have a little look. It says use information from the graph, so please don't ignore it. So he stops taking them after 4 days. So here we can see after 4 days, okay, he's still got bacteria left in there. And if you have a look, he's got a significant amount. Remember, this is measured in billions. He's got a lot of bacteria left in his body still, okay? And what would happen is if he stopped taking them, they'd reproduce, and he'd quickly shoot up here again and become very poorly again. So what we're going to say is he still has bacteria in his body, and they would reproduce and make him ill again. Okay. Occasionally, a new resistant strain of pathogen appears. The new strain may spread rapidly. Explain why. So this here would be because the antibiotic or the treatment doesn't work, okay? So maybe some resistant strains have survived or something like that. So the antibiotics no longer work, and they then reproduce because the antibiotic's not going to kill them. And now you have a population of resistant bacteria which can increase again and again and again, okay? So let's write that down. So the antibiotics are ineffective or do not work. So antibiotics are ineffective because resistant pathogens survive. These reproduce. Okay, so the population of resistant pathogens increases. Okay, so antibiotics are ineffective because resistant pathogens survive, they reproduce, and so the population of resistant pathogens increases. Okay, so that's why a new strain may spread really rapidly, because we've got loads of it and that can then pass from person to person. Okay, on to question 6. Okay, this is question 6. The picture shows a zebrafish. Zebrafish are small freshwater fish that usually have black and silver stripes. Zebrafish can tolerate a wide range of environmental conditions. Scientists have genetically modified zebrafish to act as pollution indicators. The genetically modified zebrafish have a gene transferred from a jellyfish. The gene allows the stripes of the zebrafish to change colour. Describe how the scientists produced the genetically modified zebrafish. So if it's been genetically modified, it's had its genes swapped, okay, or exchanged or changed in some way to modify it. So it's had its genes cut out using enzymes. Enzymes like chemical scissors, which can cut our enzymes out of cut our genes out of our DNA, and then we can transfer them into a zebrafish, okay, to change as we wish. So the genes have been cut out using enzymes, and these are then transferred to the zebrafish at an early stage of development. Okay, very similar to a question on the 2014 paper as well, almost identical mark scheme there. Okay, some scientists are worried about the production of genetically modified zebrafish. Suggest reasons why. Well, again, very similar to the other paper. The concerns would be that the genes could transfer to other fish species, maybe in the water. We could have an effect on a food chain if it's eaten by a bigger fish. We don't know, or humans. We don't know the effect on them. Okay, and there might also be an effect to the zebrafish itself, which might be a concern. So, 2 marks, 2 things. We're going to put down 3 now, so you're aware of the 3 comments that you could make. So the issues could be that the gene could transfer to other species. It could have the gene could have an effect on the food chain, so larger fish or even humans. Okay, and there could be an effect on the zebrafish themselves. Okay, so those 3 points would all be absolutely fine for that question. Okay, on to question 7. Okay, so the Galapagos Islands are in the Pacific Ocean, 1,400 kilometres from South America. A type of bird called a ground finch lives on the islands, and the picture shows a ground finch. The size of the seeds the ground finch can eat depends upon the size of the beak. To eat large seeds, a large beak is needed. The bar chart shows the sizes of the beaks of ground finches on one island in 1976 and in 1978. So let's have a look at these 2 charts. We've got number of ground finches, okay, and we've got on the side of both, and we've got the beak size in millimetres. Now straight away, we should be able to see that the beak size in millimetres is the same scale on both. However, the number of ground finch on this scale goes from 0 to 80, but on this only goes from 0 to 12. So there's a lot less ground finches on this chart than there are on that chart there. Let's have a look at the question. the population of the ground finches and their beak sizes changed between 1976 and 1978. Describe these changes. So one thing we've already spotted is there was far less of them in the population. And then let's look at beak size. So we can see clear difference here that there were beaks below 8 millimeters, but there's no finches with beaks below 8 millimeters in 1978, and equally there's some finches there with beak sizes beyond 12 and just under 14, but there's nothing here. Okay, so again we're going to use that as some differences. So the number's different on both, and the higher and the lower is different as well, and it's worth 3 marks, and we've got 3 points, clear points there to talk about. So what we're going to say is in 1978, please be clear which year you're talking about for questions like this so the examiner doesn't get confused. So in 1978 there was less finches in the population, and there were none with beaks less than 8 millimeters or greater than, let's be specific here, so it's bang on 8 for this one, but for this one it does creep up, so I think about 11.5 because it's straight in between those two there, or greater than 11.5 millimeters. Okay, so there are 3 points there. So less finches, nothing with beaks below 8 millimeters, and none with beaks over 11.5 millimeters. Okay, in 1977 there was very little rain on the island, so 1977 falls in between these two charts. The lack of rain affected the seeds the finches ate. The table shows how the seeds were affected. So in 1976 to 1978, and this is where there was little rain in 1977, we can see the mean number of seeds dropped dramatically. So if we've got less food, then we've got much more competition for the few seeds that are available, and we can say the mean mass of each seed in milligrams actually increased, hasn't it? That's gone up. So the seeds have got bigger, but there's less of them. So suggest an explanation for the changes in beak sizes between 1976 and 1978. Well, probably the finches down here with small beaks probably couldn't eat any of the seeds because they've increased in size. Okay, so what's caused that? Well, we know if we think about evolution, we think about mutations, random mutations, giving something an advantage and therefore more likely to survive and pass that gene on to their offspring. So let's structure that so it becomes clear. So we need to say there was a variation or mutation in beak sizes. Okay, so the ones that had larger beaks were better adapted for the larger seeds, or you could have said the ones that had the smaller beaks couldn't eat the seeds. Okay, and then the finches with the larger beaks survived and passed the gene or allele onto their offspring. Okay, so this is what happens. There's a mutation. The ones with bigger beaks were better adapted for the larger seeds. So the finch with the larger beaks survived, or more likely to survive, in that element of competition and pass on the genes to their offspring. Okay, and that's how evolution occurs. Right, on to question 8. Okay, final question, question 8. The diagram shows the annual flow of energy through a habitat. The figures are in kilojoules per metre squared. So we've got sunlight coming in to green plants, to plant-eating insects, to insect-eating birds and predatory birds. calculate the percentage of the energy in sunlight that was transferred into energy in the green plant. So here is exactly the sort of question where you're going to want your calculator. So if we're going to calculate percentage, we do what we've got divided by the total amount times by 100. Okay, so let's have a look. So we've got the amount of energy in the sunlight, okay, transferred into energy in the green plants. So these have got 10 to the power 4, 10 to the power 6. So we need to get rid of those when we're trying to work out the formula. Unless you want to type those into your calculator, that's up to you. So this becomes, okay, so 2.4 to the 4. So I think the point was there, 1, 2, 3, 4. Okay, and then we divide that by 4 with 6 zeros after it. So 1, 2, 3, 4, 5, 6. And then we multiply that by 100. So what I would do is I'd do that divided by that, press equals, and then times by 100, and you come out with 0.6%. Okay, so not a lot is going into the sun, into the green plant from the sun. Suggest reasons why the percentage energy transfer you calculated in part AI was so low. So why do we think not all the sunlight is going to go into the green plants? Well, some of that light might have been reflected. So some may have been reflected off the leaf. Okay. Some might not have even been absorbed. It might have missed the chlorophyll entirely. So remember, the chlorophyll contains a chloroplast that absorbs. So some may not have been absorbed. Okay, so that's already worth 2 marks. The other things you tend to get marks for is it might have been the wrong wavelength. So green plants absorb all other colours apart from green. So if it's the wrong wavelength, then the plant wouldn't have been able to use it. Okay, also the plant may have lost some energy as well. But these are the key things in terms of how the sunlight isn't getting into the green plant. Reflection, and some of it may not have been absorbed by the chlorophyll in the chloroplasts. Okay. Compare the amount of energy transferred to the insect-eating birds with the amount transferred to the predatory birds. Suggest explanations for the difference in the amount of energy transferred to 2 types of bird. So we want to compare the insect-eating bird and the predatory bird. So the predatory bird is getting 15 kilojoules per metre squared, so that's 15. And the insect-eating bird is having 200 transferred to it. Okay, so we'll put that on there. So remember our numbers. Okay, so you need to think about why these two are getting different amounts transferred to them. Okay, so from this bird to that bird, this insect-eating bird is going to lose energy. Okay, so the insect-eating bird will lose energy, and we now need to give some reasons about why it would lose energy. So it will lose energy in feces or waste, you could put. That would be absolutely fine. If you can't spell feces, there's a funny word to spell, so you could just write waste. And it may also lose it in respiration or movement. Okay, but they're often given the same mark, so don't write just those two. You need to put waste and respiration or movement. You might also find that the predatory bird doesn't eat all of the insect-eating bird, whereas the insect-eating bird probably eats all of the insect in one go. I should think the predatory bird wouldn't eat all of the insect-eating bird. So let's carry on with that one there. So the predatory bird will not eat all the insect-eating bird, but the insect-eating bird probably ate most of the insect. Okay, so that's that final question, and then we have finished this page. paper.